Professor in the Computer Science and Engineering Department of Jadavpur University. He has more than 22 years of professional experience. He received his PhD from Jadavpur University and did his postdocs from University of Iowa, USA, and University of Warsaw, Poland. He has worked in many reputed international institutes and has more than 300 research publications in the area of pattern recognition, machine learning, bioinformatics, etc. He is the recipient of Research Award from UGC, DAT Fellowship from Germany, Visiting Research Fellowship from Japan, European Union, France, UK, and Fast Track Fellowship from DST Government of India. Now I am requesting Chair to continue the session.
Yes. Tak, mówisz teraz o tym był. Bo tam myślałem, żeby sprawdzić CTCF versus RNA-POL2 dla tych 4 linii. Tak. I to jest wtedy nawet nie review, a to jest taki idea paper bardziej. Niby to nie jest długie.
Tylko przegląd literatury, tak? No to jest papier z empatii, jak rozumiem.
Znaczy wiesz co, znaczy rozumiem, że Abiszek powinien wziąć ten skrypt i porównać CTCF-owy sygnał z RNA-POL2. Tak? Znaczy wiesz, bo to ciapety, no to jest tylko wziąć test for DNA, puścić skrypt i już są gotowe tabelki. Tylko tam jeszcze jakieś konkluzje trzeba wyciągnąć głębsze, ale...
Przeglądówka, więc już tak nie wiem.
A może wiesz co, a może wziąć ten review, to Frontiers, dodać ten twój wkład.
Wkład
to tu, prawda, tylko tak, bo ty robisz przegląd literatury w jakim temacie tych grafów? CTCF i RNA-POL2.
I nie kohezyna.
Kohezyna. O, no to nawet to i pokazać, można wziąć dla tych 4, co mamy CTCF-a z Fordiana, żeby Abiszek przesłał.
No i face. Hello, do you hear me? Do you hear me?
Yes.
Yes, okay. Okay, so Thank you, Professor Dariusz Plewczynski, for kindly accepting this invitation to deliver a keynote lecture at the fag end of the day in India, and sparing some time from your busy office hours in Poland. So before we start the session, it's my honor to read out your brief bio. So, Professor Dariusz Plewczynski, after obtaining a master's degree in theoretical physics in the Faculty of Physics in the University of Warsaw, began working in 1996 as an assistant in the Institute of Physical Chemistry of Polish Academy of Sciences. After defending his doctoral dissertation on June 25, 2001, he obtained a PhD in chemical sciences in the field of physical chemistry. Then he started working at the Interdisciplinary Center for Mathematical and Computational Modeling, ICM, of the University of Warsaw, and on October 11, 2012, he obtained a postdoctoral degree in the technical sciences in the field of technical computer science, specializing in bioinformatics. On December 1, 2015, he changed his place of work at ICM with ICM, moving to the Center for New Technologies, CENT, of the University of Warsaw, and then on October 1, 2017, he started working as the Faculty of Mathematical and Information Sciences in the Warsaw University of Technology. On July 21, 2020, he was awarded the title of Professor of Exact and Natural Sciences. His achievements are an example of exemplary performance of the tasks of researcher with particular emphasis on interdisciplinary research. The publication achievement of Professor Dariusz Plewczynski includes 118 peer-reviewed publications from the Philadelphia list, including 19 publications in the last 2 years, 2019-2020. His achievements concern the research areas in engineering and technology sciences, technical information technology and telecommunications, exact and natural sciences, in biological sciences. The number of citations only among documents included in Scopus database, excluding self-citation, is 6152. Professor Hirsch in index, Dr. Dariusz Plewczynski is 23. So an important element of exemplary performance of duties by Professor Dariusz Plewczynski in the organization of research and participation in international cooperation. Dr. Dariusz Plewczynski is currently managing 2 4-year research projects funded by the National Science Center and the team project with FNP, participating as a consortium member or co-leader in 4 international projects. With this, it's over to Professor Dariusz Plewczynski, and thank you again.
Thank you very much for a nice, kind introduction of myself to the audience. I'm very sorry that I could not join you in person, so I'm only in the virtual mode. So that's how life sometimes looks like. And today, following suggestions of the organizers, I would like you to take for a small tour. The Polon, as the title suggests, but it will be actually a tour over genomics. And the genomics is the recently developing field that somehow fuse bioinformatics approaches with the wet lab molecular biology experiments in order to understand life, basically. So that's the goal, and maybe I will start, not to, because I know that there is some delay, so I will try to even shrink a bit my presentation. I hope I will manage to do it. So let's start. Okay, just to make sure that we understand the term Tour de Polon before we will go to genomics. So as you know, Poland is located in a small part of the world, which is Europe, and is actually more or less in the center of the Europe. And the Tour de Polon is actually south of Poland, international biking competition that is, at least in Europe, very famous. So anytime you visit Poland, it is worth to plan that somehow around beginning of August, when you can actually participate and as a and observe people running and trying to reach the end point as the first ones. Okay, so, and the reason I use this title is that I will not be only talking about structure and functional genomics. I will start with that, but I will be talking also about team, about the laboratory. So our SFG laboratory is actually the laboratory which fuse work of many people actually involved in the study. And this type of teamwork is actually something that I was exposed while being in US, and therefore we are trying basically to implement something like that here in Poland. Okay, so we have two parts. If I will, I will try to make it like more or less short, not to cover too much time, but generally I will start with the introduction. As far as I know, most of you have not been exposed previously to genomics, DNA sequence, or some bioinformatics genome-related topics. So I will try to is some basic concepts, and then, as I told you, Tour de Poland, so the laboratory that actually is working on the 3D genomics topics. Okay, so let's start with the introduction. Just one comment: the flag here is a Ukrainian flag. As you know, in Ukraine there is presently a war, which actually was never expected, at least in the Europe, to happen, and that's why actually part of my laboratory are people from Ukraine. So that's why we somehow would like to honor them and the honor of Ukrainian people, Ukrainian country, for defending freedom against the Russian invaders. Okay, so that's more or less the political comment before I dive into pure science, which is apolitical. So okay, so let's start with the basic concepts. As you know, genomics is strongly founded on certain observations which were actually gathered from the last more than 100 years. So we could think, based on what we know now, that actually the most of the new informations that we know about DNA is about its sequence. So the sequence of nucleotides that form a long chain of DNA double helix. But actually, if you go to history, everything started with the structure, not a sequence, but the structure. So physical, three-dimensional objects that biologists actually observe under the microscope. So those features here show basically in the end of 19th century, people observe under the microscope that cells, while dividing, form certain strange, mysterious structures that are being divided during the cell division. So that was something very striking and suggesting that there is a specific biological or biomolecular medium that carry the information, okay? Because it was supposed that biological information is stored in a cell, in a certain object, certain physical biological molecules, and then during the cell division you have to divide the information. So basically that's the process that they observe of division of the cells, which, apart from dividing the cell into two parts, also divide the information, okay, into two copies, basically. Okay, and then of course, I mean, before the sequence somehow exploded, DNA sequence analysis exploded, people realized that the molecule that carry the information is actually a DNA, and it's formed double helix. So those are, physical famous X-ray photo that actually based on this X-ray diffraction, it was discovered that DNA form a double helix, and the obviously very detailed mechanical model was proposed by Crick and Watson. You are probably familiar with the whole story. Apart from micro scale, people in around 75 discovered that actually what is really inside the cell is not pure DNA double helix. Actually, double helix is somehow attached to many proteins, and this biomolecular complex, which is DNA double helix plus the proteins, is actually what it is called chromatin, and it is actually, you can observe them under the microscopy. You need to go to high resolution, so it's not that it was done previously in a light microscopy. You have to have like electron microscopy for that. Okay, so when talking about passing the biological information, then we have to talk about the structure. But obviously, what we all know probably by heart is that everything is coded in the DNA sequence of base pairs. Okay, and therefore one could put suggestion that DNA sequence defines its biological function, and biological function of this DNA sequence is related with the all cellular states that you can define for simple organisms, more complex organisms, or even higher organisms such as mammalians and humans, for example. So this concept that we have to understand the sequence was actually founded and explored starting from around 2000 year, where people start to sequence not only a single individual from the population of, say, humans, but start to sequence more and more humans. Okay, and of course, the first human genome project DNA sequence of a human individual was a single person actually, and it was published in Science and Nature, and it was a great breakthrough because it was the first time that a DNA of human being was sequenced. So it was read by very extensive experiments, and it costs huge amounts of money actually. But and it was on a single sequence. Therefore, you probably are familiar with the concept that each species are being characterized by the so-called reference genome sequence. So you can say that around 2000, actually, we discover a human DNA reference sequence from those experiments. But very quickly, we realized that actually, if you take more than one person, for example, a family, okay, three persons, mother, father, and daughter, or a son, then you realize that between father and mother there will be some differences in DNA sequence, which seems to be like more or less obvious now, but that time it was under several discussion. What is the extent of differences between humans? And the 1000 Genomes project that I actually was participating discovered that the variability of DNA sequence among many human individuals forming our human population is actually very high. It's not low, it's very high. It's not that the differences between us can be refined reference like a couple of mutations. No, it's actually millions, tens of millions of base pairs that different any two individuals taken from the human population. And it was the huge discovery. Okay, so people realize that within human population, and that it will be the same for a different species like mouse, rat, yeast, any species you take, there will be some variability of DNA sequence. And obviously, due to this variability of DNA sequence, there will be some differences in a phenotype. So how, in the case of human population, how the person looks like, which disease, which risk of disease is actually being observed for a given individual, and so on and so on. Okay, so that was the huge step forward in understanding our the link between sequence and biological function. And shortly after, next breakthrough was proposed that defined that apart from DNA sequence, so the list of letters, each letter describing one type of nucleotides. So we have 4 nucleotides, A, C, T, G, and then there is like a long set of those letters forming DNA. So people realize that because of this very concept of actually chromatin, it is not only the sequence, DNA sequence, which matter, which carry the information. Apart from DNA sequence, so mutations of those base pairs, what we observe that also the state, so the post-translational modification of proteins that are binded to DNA sequence also influence or carry some biological information. Okay. And this is important because it explains the differences between various cells within a single individual. Okay, so if you take a single individual, you see a lot of different tissues, and each tissue, obviously, it is the same person, so has exactly the same DNA sequence, with certain approximations because during the development, growth of the organism, and then life of a given individual, we accumulate certain mutations. So the DNA sequence can be also a bit changed in different cells forming a given individual. But generally, the differences between various tissues are not due to the more or less the same DNA sequence across all the cells forming a given individual, but due to epigenomic factors, so the basically reversible states of proteins that additionally code some additional information, which is tissue specific. So there will be a different set of activated or, for example, phosphorylated histones in the case of neurons, and there will be different histones, different locations on DNA sequence covered by some methylated histones in the case of skin or B cells. Okay, so that is the crucial concepts that are both the DNA sequence and the epigenomics are linear information, okay, because they are assigned to the position on a DNA double helix. Okay, so we have the sequence information, so one-dimensional information that is coding either the base pairs, so nucleotides, or the epigenomic information, but we have also 3D structure, as I was describing a moment ago. So we are actually participating in the 4D nucleome project, and the 4D nucleome project is a huge invader, similar to the human genome project, that with the aim not to find the sequence of a human individual, but rather understand and describe the structure, three-dimensional structure of a human. Okay, so we are applying many different techniques within this huge consortium, and for example, you can actually see that there is some, obviously, sequencing effort that identify those letters I was talking about, ATGC. Okay, so those are nucleotides forming this double helix. But apart from them, we have certain proteins that are binding to DNA, and together they form chromatin. Okay, and the characterization of where those proteins are binding to DNA, that is discovery of the DNA sequence motifs that are recognized by those proteins, and also changes, post-translational changes of the structure, influencing the structure of those proteins upon post-translational modifications. They are also important and responsible actually for the three-dimensional structure of chromatin inside the nucleus. I will be talking about that in a moment. Apart from experimental techniques, there is functional validation, which is extremely important. And you are probably familiar with the CRISPR technique, which allows you to modify DNA sequence. So to remove a part of the DNA sequence and observe the effect of the living cell when the certain deletions or inserting some piece of the DNA sequence into a given location, how it influenced the living cell. Okay, and we have the important part of the whole story, namely, apart from molecular biology, functional validation, we have an important part of the work, which is basically machine learning, polymer biophysical modeling. So generally constructing information-aware models, computational models that can explain the experimental data, basically, and the functional effect of some changes that we introduce into the living cells. So the FARDINUCROM project is tightly linked with epigenomics because it is not only the DNA double helix, okay, it is not only DNA sequence, it is also proteins, okay. So epigenomics is very relevant in describing or in explaining the 3D structure and the biological function of certain regions of the DNA. And secondly, as it was shown in the ENCODE consortium, most of the DNA that was actually not, it was previously discarded because people focus on the coding fragments of DNA, apparently was proved to be responsible as well for a biological function. So non-coding regions of DNA, which are actually the very large part of the whole DNA sequence, are also important to understand biological function. So the generally the goal was building like a GPS for the three-dimensional genomics. So in the case of GPS, you locate yourself on the map of the world. That's why the maps of the whole world and Europe and Poland. But in the case of 3D genomics, you would like to also locate where you are. By you, I mean the certain fragment of DNA sequence, where it is located in 3D space of the nucleus. Okay, so that's a challenge. And if you are at least generally familiar with bioinformatics, you probably remember from the basic courses on bioinformatics paradigm of sequence that actually is responsible for the structure, and the structure is explaining the function of a protein. Here we have similar approach. We have a DNA sequence. We have the structure of chromatin, so how the DNA sequence is put into the forming certain three-dimensional objects that we can see by, for example, microscopy. And the structure of chromatin explains the genome biological function. Okay. So obviously, because DNA is a complex molecule, it is a multi-scale problem, and I will explain it in the next slides. So first of all, as people discovered, we have DNA, as you know, DNA double helix. Okay, so its size is like around 2 nanometers, and it's basically A, C, A, G, T, and so on. Okay, so this is the sequence of the letters. Then this double helix is going around the nucleosomes, and nucleosomes are protein-protein complexes formed by histones. So the octamers of histone proteins that form certain like cylinders or balls in this representation. And so in order to pack the DNA, it has to be intertwined around the nucleosomes. The size of the nucleosomes will be around 10 nanometer. Then it will form some higher order structures, such as, for example, the chromosomes during the cell division. So this is exactly the whole chromosome is what people at the end of the 19th century observe under the microscope. This is macroscopic objects, okay, because light microscope you can actually observe and then see by yourself, built out of the very tiny object, very thin double helix. Okay, so that is the multiple spatial scales. And if you go like deeper into understanding how this DNA double helix and the nucleosome, so the chromatin, how it folds into this beautiful large object, which is the nucleus, then actually you can identify multiple distinguished spatial scales, such as chromatin loop, compartments, and chromosomal territories. And I will be talking about that in a moment. For now, just one comment here that loops are objects, three-dimensional shapes. Okay, those are 3D structures composed of around 10,000 base pairs. Okay, so I take the double helix, I take 10,000 of the base pairs, so 10,000 of the letters, and I form like a loop, and it looks like more or less like the loop. This is actually here it's not double helix, but it's a chromatin. So actually it is this part which has a size of 10 nanometer, and it's represented as this thin fiber on this picture. And to the chromatin, CTCF proteins and cohesin proteins are binded, and they are responsible for this specific shape. So for forming actually the chromatin fiber. CTCF protein recognize a specific CTCF binding motif on the DNA sequence. Therefore, it knows where to bind. And when binded to DNA, it forms a dimer, okay, with another CTCF binded to another place of the DNA on the chromatin. And then cohesin ring, which is another protein, binds to CTCFs and stabilize the complex. Okay, so those chromatin loops are very abundant across, and they can be visualized on the heat map, which represents basically the distance between some kind of estimation of the physical distance between various base pairs, basically on DNA to all other base pairs in a given fragment. Okay, so if I take this fragment of 10 kb of the base pairs, so 10,000 base pairs, I divide it into so-called resolution 5 kb. So that would be each fragment over here will have the certain size. Then such loops basically can be visualized on the heat map as a dots. And I will talk about that in a moment in more detail. So those heat maps are observed. So those are experimental data, and they reflect different spatial scales, such as chromatin loops, TATs, so topologically associating domains, compartments, and chromosomal territories. And I will explain it later. For now, it is important to see that the resolution, so the scale, is being defined by the size of the DNA segment that folds into a specific shape. So the chromatin loop will be around 10 kb and more. Domains will be of the size of 1 million or 2 millions of base pairs. So there will be very large, actually, fragments of DNA forming like a hairballs, which you see over here. So we have genomic domain 1, 2, 3, 4, 5, 6, okay, over here. And those domains are further divided into so-called compartments, which reflect the activity of the DNA. So which parts of DNA are being actively read by some certain transcriptional machinery. Okay, so we have active compartment, inactive. And then we have chromosomes, okay? So those chromosomes that I showed here and the previous slides, those are the whole chromosome 1, chromosome 2, and so on, that tend not to mix to each other. And I will show some proof that they are really not mixing with each other in a moment, okay? And all those structures with different scales are being reflected on those distances, distance maps, as a certain motifs, which are spatial motifs. So you can think about those heat maps as certain images that you can process by machine learning or image processing computational algorithms to, for example, find those dots, okay, which represent single chromatin loops. If you have image processing algorithm that can find such a dot, then from the experimental data, which is the heat map itself, you can find all the loops that are present in this region of the DNA. Okay. Okay, so we have definitely multi-scale 3D structure of chromatin inside the nucleus, but we have something more, because I was telling you about the differences in DNA sequence. Similarly, we see here that modifications of the sequence, so by CRISPR technology, for example, we can, for example, delete this fragment of DNA sequence, which is here depicted by the box, and then, okay, we remove this part, which is basically over here, and we see how the structure of chromatin is changing. So you see that if you remove this so-called boundary element, then this part of the chromatin structure fuses with this part. So you have large, one single fused genomic domain. Whereas over here, you have domain 1, domain 2, domain 3, okay, which is reflected on a heat map of all against all distances, like 3 triangles. This is domain 1, this is domain 2, this is domain 3, okay? And after deletion of this small element of the size of, say, 1 kb maybe, okay, with the CRISPR technology, suddenly you realize that no longer you have 3 domains, you have only 2 of them, okay? Domain 1 stays untouched because there is still a boundary element, but domain 2, because of the deletion of the boundary, fuses with domain 3, okay? So you end up with a single domain. So there is definitely the difference in 3D conformation of chromatin when you change the DNA sequence. And this is important because now you can imagine that you're analyzing different DNA sequences from different individuals, human individuals, and therefore each deletion or duplication or some changes of the DNA sequence influence actually the 3D structure, okay, of the chromatin. Okay, we are talking about differences between different individuals. What do we mean by those mutations? Okay, what is the definition of those mutations? So apart from single nucleotide polymorphisms that we take, for example, 3 individuals, we sequence those 3 individuals, and then we realize that, for example, individual 1 has A in a certain position, okay? Individual 2 have G, then we have T. So this is the example of the single nucleotide polymorphism. Across a population of humans, we have the difference for this position on the nucleotide. So those are short ones, a single nucleotide changes. There are a bit longer ones, so-called indels, so insertions and deletions, short insertions and deletions. They are below actually 50, yeah, okay, so that's 50 base pairs that they are very short, and they actually comprise of multiple nucleotides. And most of the variability among the human population is due to larger so-called structural variants, which are longer than 50 base pairs. Those are insertions, deletions, inversions, duplications, translocations, okay? Each of these type is basically a different class of the mutation that you can meet by analyzing a human population. Okay, so we have a sequence variability. It was proved by 1000 Genome Project that this sequence variability is enormous, even across human population, even across the same population, subpopulation. The variability will be enormous. It's not that, for example, Yoruban versus European, they have a lot of differences. No. It's actually within Europeans, you have more than 20 millions of the base pairs being affected, not affected by being different from any two individuals. And as well in Yoruban tribe in Africa, they will have also variability. So the variability is not linked with the subpopulation. There are some subpopulation unique structural variants or SNPs or those, but actually majority of the variability is everywhere. Okay, it's not related with the subpopulation. Okay, so we have those changes in the DNA sequence. And what about a genome spatial of conformation? So as I told you, there are chromosomal territories, and it was proved actually in 2005 and then 2015 that chromosomes do not mix with each other. Okay, so if you take a set of human cells and you paint under the microscope with a specific probes different chromosomes with different colors, then actually what you discover is a beautiful rainbow of different chromosomes that do not mix with each other. If those chromosomes will mix with each other, then the outcome will be not colorful, but it will be like a gray color all around. Okay, so by using 24 color 3D fish, you can actually prove that chromosomes do not mix with each other. Okay, that's extremely important conclusion. Moreover, you can see that there are compartments, and the compartments reflect activity, either active regions of DNA, those are the green, and inactive ones, which are typically close to the nuclear membrane. By going deeper, actually, you can identify the chromatin loops and chromatin regions, basically the topologically associating domains. They are formed by many protein factors. So again, to understand genomics, you have to not only analyze the DNA sequence, you have also to understand proteins. Okay, that's important point. And finally, we have these chromatin loops that I was mentioning before that are responsible they're involved in transcription, replication, and there is our CTCF protein I was mentioning, which binds to the CCCTC binding motif. Okay, so that's our knowledge, and then we have certain experimental methods, either light microscopy, so the same methodology even from the 19th century. You look through the microscope and you try to see where actually certain regions of DNA are located, and by painting it with the probes, colorful probes, you can actually select certain regions of DNA and observe the exact 3D position, XYZ coordinates for those. And you have next generation sequencing methods that produce the heat maps I mentioned earlier that reflect all against all, basically physical distances between all locations of DNA sequence. And from this second part, actually, the two methods of choice are either Hi-C or ChIAPET. Hi-C provides the heat map I mentioned before, and in the case of ChIAPET, you have set of loops, so you actually observe only chromatin loops. Okay, only chromatin loops. So that's basically the summary. From the Hi-C, you can, by analyzing heat map, so all against all distance map of different locations of DNA, you can actually find those topologically associating domains, so genomic domains. And moreover, you can find those dots, which represent chromatin loops. Whereas in ChiaPET, basically, you get certain experimentally processed information, which directly give you a set of chromatin loops. And you can compare those techniques. They basically agree with each other. Okay, and the agreement, as you see, it is not like in the pure mathematics. Actually, in biology, agreement with the correlation 0.7, 0.6, it's a very good correlation, actually. So that's something. It's not precise. Always biology is very imprecise. Therefore, you need to actually really be permissive in terms of the correlation values because of many factors, experimental errors, and so on. Okay, so with those experimental methods at hand, we can start to understand the biological function of DNA and also details of the three-dimensional structure of DNA when it's folded into a single cell nucleus. There are some problems, and experimental data requires a lot of interpretations, and those a lot of biases is due to the nature of the system, but also nature of the experimental methods. For example, a couple of examples: GC content and mappability of DNA sequence. So how many GCs you have in a DNA sequence, mappability, how complex the DNA sequence is in the given region. They all influence your ability to observe those fragments, because the technology, next generation sequencing technology, has problems with, especially in the case of Illumina technique, with the low complexity regions, because those are short fragments that are difficult to be mapped on the genome. So you have to be aware about those biases. Also, even if you have set of interactions, set of three-dimensional interactions, you are not sure that if they are really linked with except from ChiaPET, if they are linked with a specific protein factors that are mediating those interactions between two locations on DNA double helix, or maybe they are just random ligations of the regions close by. There is some influence of the lamina. And finally, the most difficult problem to tackle, it's actually the population average. So the experiments that are done actually are collecting data from 100 millions of cells. So the representation, the 3D structure that you see, it's like average over all possible conformations that can be observed in a living cells. So you never know if this is the 3D structure that is actually in every cell, or maybe in 50% of cells there is a single chromatin loop, 20% there is another loop, 30% there is another loop. If you sum over that an average, you have this population average. So it's like rugby player that you average many photos of the dynamical rugby player running around, and then you get something which is really difficult to interpret. Okay, functional impact, very important. I just mentioned that those deletions I mentioned that you could introduce by CRISPR, they are happening in nature, okay, by some random mutations. And those mutations that affect the 3D structure can have an extremely striking impact on the development, both in terms of structure variants, but even the single mutations, single SNPs can influence the chromatin loops by influencing the CTCF binding motif. And the challenge that we are facing in the laboratory is to predict those spatial motif, spatial features of chromatin using sequence and experimental DNA sequence and experimental data. So again, just to keep in mind, we have loops, we have lamina, we have domains, we have compartments, we have many different spatial and temporal scales, and we have a bunch of the methods that we developed in the laboratory that are either polymer-based, restraint-based, or machine learning-based that exploit also the epigenomic information apart from DNA sequence. So computational modeling, the modeling that we are doing in the SFG lab is based on experimental data. So it's like a magic box. You put a lot of experimental data that you collected in the wet lab, and actually we are doing the wet lab experiments in our laboratory. And then from this experimental data, we infer the 3D structure and at different resolutions, okay? And the typical approach is you start with high-C or CHIAPET data, you randomize the interactions, you build the 3D conformations that reflect each of the possible sets of chromatin loops, and then you again average over all possible conformations, and you check if you have similar heat map like in the experiments. And the approach that we are implementing is based on a multi-scale Monte Carlo. So it starts with the global conformation. Then we model different scales going to the different and higher resolution. Okay, so those are a couple of examples I just want to show you that basically show that based on experimental data, you can reasonably build a reasonable 3D structures that reflect what we see basically in next generation sequencing, okay? And the models, this is, for example, location of the center of mass of different chromosomes. And this is the final multi-scale model of the nucleus for GM1278. Going bit toward the imaging that I started with the actually 3D fish and the light imaging, the concept is slightly different. Previously, it was from next generation sequencing. We try to predict the structure. Here, we observe something. So we see the structure, for example, in such nice three-dimensional images that we collect with our collaborators. And this is the example of this multicolor where each color actually reflects the different chromosome. So there are many challenges that are linked with interpretation of these experimental images because you can go for, this is macro resolution and this is micro resolution. So this is the electron microscopy image of a single genomic domain. And this imaging data provides a very important validation for the polymer models that exploit biophysical modeling or machine learning because they are independent experimental data that are not used in construction of the computational model. Okay. Of course, there is like a bunch of problems if you want to process, this is different nuclei, and you need to analyze very weak images sometimes, so you have to have a lot of actually computational methods actually to process the imaging. Those are chromosome 1, human chromosome 1, observed as you see the chromosome 1, there is one copy, another copy, we have 2 copies of chromosome 1. And there is a lot of variability across those are different nuclei, different cells, and you see the differences in the volume, shape of the chromosome 1 across different tissues. This is definitely macro resolution, yeah, so we use that. Maybe I will not go into details from the lack of time. We used different techniques which are based on the quad tree optimization and influence maps. And basically what we discover is that if you use constraints coming from imaging, then you can construct reasonable 3D models that are consistent with the, at least in the macro scale, with what is observed under the microscope. In the case of meso resolution, so the resolution of single chromatin domains, the methodology starts to be more complex. We use molecular dynamics, and we can basically reconstruct the chromatin folding. And maybe I will show you how this is each color marks different chromosome. They are put into the volume of the nucleus, and then we run molecular dynamics. Maybe I will speed up this is original image, okay, and we put into the original image the multiple chains representing each chromosome, and then we run molecular dynamics in order to fill the image. So this is the way how we deal. So we start with the polymers, color represents different chromosome, and then we exploit image to guide folding of chromatin in order to get the final and correct conformation of the chromatin for a specific image. And finally, micro resolution. This is we use several methods well known from the computer science, skeletonization of the objects in order to build reasonable models of the genomic domains and use the imaging data as a validation for our biophysical modeling. Okay, so this is example. This is original ChiaPet, and this is the heat map that we get from the simulations, and we can compare this heat map with the original data, but also we can validate it comparing with the images. Okay, so this is another example of electron microscopy. Electron microscopy has to be processed, so it has to be segmented, and there is many algorithms that are used to segment the 3D images. And then we can start to perform a modeling. In this case, it was multidimensional scaling as the engine for the modeling. And we get polymer models that are presented here that are consistent with the image. Okay, similar approach. This is electron microscopy. Similar things can be done with IPALM, which is super resolution light microscopy. So we are able to use imaging data to actually build the reasonable polymer computational models. Highest resolution is a single chromatin loop. Okay, so this is the loop I was mentioning, to CTCF cohesin. And this is how it looks under the microscope. So you see that it's extremely difficult to see actually where the chromatin loop is located. So basically that requires developing an algorithm that could model, that could simulate, that could read from this experimental image, three-dimensional image, where actually the chromatin loop is located. And we do it by finding the centers of the light and then connecting those dots by the traveling salesman algorithm, and then we end up with the three-dimensional chromatin fiber that is actually observed under the microscope. And then we can compare with the genomic data. Okay, we have several methods to compare, to construct the chromatin models, and then we can see what is the similarity and difference. This is the imaging. This is the imaging 3D model, and this is the model that actually represent what we see in genomic data. Okay, so actually the similarities is pretty high in this case, and this is a single chromatin loop. So the single chromatin loop that we observe in the genome. Okay, speeding up because my time is passing by. So from sequence we can predict structure, and this is the 3D GNOME web server that we published recently. The link is over here. You can go to the web page and actually observe. I will not go into details. Maybe for the next time I will go into details. But generally, it implements the molecular multi-scale Monte Carlo method. And in this engine is a web server, so you don't need to install anything. You just go into the web link, and you can perform build your own chromatin folded DNA models, and actually for a specific locations. Okay, so the part one actually take me longer than I expected, so I will probably wrap up the part two in a moment. But generally, I would like to stress that I was talking about the major challenges that we face in structure and functional genomics. Okay, so those challenges are still unsolved, and there is new experimental methods coming, and sometimes it happens that a new experimental method, which is more refined, more accurate, somehow change our way of thinking about the genomic, how the 3D structure of the chromatin is being observed in for example, human cells. Okay, the part 2, it's about the laboratory. So I promised to tell a bit about our laboratory. So we are located in 2 places, University of Warsaw and the Warsaw University of Technology. And this is like not all actually lab members. We are actually first located on the University of Warsaw, and I would like to mention Kaustav Sengupta, who is a PhD student there, and also postdoc Karolina Jatkowska, who is working in a wet lab. So in the University of Warsaw, we're actually working both on the wet lab and bioinformatics, whereas the Warsaw University of Technology, there is like mostly bioinformatics. I'm still not updated Anup here, so I hope I will do it in the next occasion. So this is purely bioinformatics and computational genomics laboratory, and we are equipped with the advanced computing HPC cluster. Okay, so just quickly going to for some conclusions. I was describing to you the multi-scale nature of the biological information that you can observe in the DNA. So the biological information that defines what is the phenotype either at the level of the single cell, which type of the cell, which is the state of the cell. Is it in a healthy cell, or it is actually influenced by some disease? It is coded in the DNA. Okay, so we have DNA, double helix, then it is intertwined on those nucleosomes. Then it forms chromatin loops. So we have another scale, spatial and temporal scale. Then those are genomic domains, AB compartments. So this is non-active chromatin, this is active chromatin. And then we have these chromosomal territories that I was showing to you, beautiful pictures from the 3D fish. The goal for all those works are actually investigation of some fundamental biological processes. So how the linear DNA sequence influenced the state of the cell, or going further, the state of the organism. How the three-dimensional chromatin structure influenced the function. Okay, and the genome function is transcription, is the repair. Sometimes DNA double helix breaks down. There are some breaks, and you have to repair that. DNA replication. So when the cell division is observed, as I was mentioning on the first slide, the biological information is being copied. So this is the application of DNA, basically. And the applications of all those challenging research questions, methods, experimental methods, computational methods are enormous. And I would like to mention genomic medicine, clinical problems. Those are the typical challenges that are actually observed. And we are using DNA sequence, epigenomic information, and the 3D structure of the chromatin to explain basically the mechanisms behind different diseases. And I already proved to you that it's not only genomic next generation sequencing data. What you need is you also need microscopy, you need epigenetic markers, and the observed changes propagates actually from the local changes of the sequence through the genomic domains into the function. Okay, and the critical point here is that the basic unit of spatial interaction in the human genome, and generally in mammalian and many other species, is a chromatin loop. It's a binary interaction that connects two far away located regions of DNA. And if you are familiar with the theory of knots and knotting, then obviously you can ask a question, okay, I have those loops, but maybe what those loops are actually forming are topological nodes. And this is examples that actually we are working on this problem. It is more mathematical problem, but you can actually appreciate that two chromatin loops can be separate from each other. So what is happening in loop 1 is independent of what is happening in the second loop, but when there is a link, there is there is certain interaction between those two loops. So the communication starts to be between AC regions and DF regions of the DNA. Okay, and you can generally represent, and this is the hot topic now, at least in our laboratory, how to represent chromatin structure or chromatin interactions in terms of the graphs. Okay, and this is work of the Kaustav Sengupta, who is also working on the interpretation of single nucleotide polymorphism, so those single nucleotide mutations that I mentioned before, and structure variants, how they influence the biological function. And this is the thing we initiated a bit of time ago. Maybe I will skip the examples if I allow, because my time is, I believe, ending. I would like to refer to another important part of the story, namely that apart from observing and representing by graphs, basically by networks, which chromatin loops are being observed in experimental data, we can go further. We can predict actually where those loops will be formed. Okay, so, and this is I would like to end maybe at this point, but I would like to mention that with the concept that shows that chromatin loops, so binary interactions between two remote regions of DNA, and you see here the linear DNA double helix location. This is like one fragment of some random chromosome. You can observe and represent chromatin loops by those arcs, and then translate them into a spatial model that could be either the biophysical polymer model or the spatial graph model. And all those three-dimensional structures, chromosomal territories, genomic domains, can be explained by these simple binary interactions that are chromatin loops. And those binary interactions, as I explained to you, are linked with some specific proteins binding to specific regions of DNA. And this specificity of binding of a protein to a DNA sequence, it's something which is ready to be used for deep learning technologies. And this is work of Anup Halder, developing and training of deep learning methods on all the plethora of the universe of the experimental data sets that actually observe certain interactions in specific regions of DNA. So somehow by observing social network of the DNA segments, we are trying to build machine learning predictors that will tell us which fragments of DNA can communicate with each other. Okay, and this is critical to better understanding biological function at the scale of the whole genome. And that's all. So thank you. I was planning to extend the part 2, but I think that I was asked actually to make it like maybe more introductory, to introduce you with the basic concepts of the genomics, three-dimensional genomics, before going into like more detailed studies. So thanks a lot.
Hello. So, thank you, Dariush, for really interesting and really covering almost like your total 3D genomic study on an overview side, starting from that the chromatin lobe structure. And I was really I was actually curious to know a bit on that your aims on the therapeutic and diagnostic use. Like, since you are talking about more on multi-scale structure. So do you think that from the sequence to structure to function, so can that be extended to more on a, from the therapeutic side and also on a diagnostic side, so that in the era of like personalized medicine, so can this study be extended up to like kind of I know that you are also into drug design and docking studies, so can that be related in the downstream, like starting from the genomics to the personalized medicine? So I was just curious about this. I know, like, we can discuss later, but if you have any quick comments on that.
Yeah, so that's a very interesting actually question, and we are actually working now extensively with Kaustav Sengupta and Anup Halder on the models that or the certain concept that tries to extend the chromatin interactions and the differences or mutations in the DNA sequence, how they can be translated into some changes in the on the level of proteins. So how from the genome somehow predict the differences in the proteome. And the proteome is basically the cell machinery that are actually responsible for biophysical interactions and biophysical processes. So we had actually a couple of examples of the functional SNPs that are happening on the level of DNA sequence that could be translated by this framework of multi meta networks. So multiple networks, one for genetic information, then for the metabolic, for the proteins level, gene ontology and function. And I will go into details, but generally, this is the vehicle that we would like. I like this slide by Kaustav, prepared using some online. So basically, from the differences in your DNA sequence, what is actually then you would like to interpret an mutation, you would like to interpret it on the level of the proteins and the biophysical processes. So and for that we are constructing spatial graphs and basically analyzing many different. Actually, those are very impressive numbers that the whole team, Kaustav, Anup, and others in the lab, managed to actually identify many of SNPs, so even single nucleotide mutations that are related with different diseases. And by analyzing the chromatin structure, actually we find many of the situations that where actually those changes, either single nucleotide like here or the structure variants, that they influence. Maybe I will go to this slide, that actually can be interpreted in terms of the given disease. So definitely that's our goal. But I agree with your comment that presently I would say those two words of proteins and the word of the whole genome sequences are a bit separated from each other for multiple reasons. So there is urgent need actually to bind them together, actually to have a clear bridge between various mutations that we observe on the level of DNA sequence and the functional impact, which can be explained in terms of biophysical processes. So to build this bridge, I believe the approaches that use meta-networked computational technique to represent the basically biomolecular interactome is actually the best solution, at least at this point.
So, Derek, we have one question from live stream in YouTube. So, do you see the question at the bottom?
Not really. If you can.
Yes. So, question from Avira De, that she's asking like which molecular dynamic software was used in this study? Is there any specific molecular dynamic software available for genomic study?
Definitely, yes. There is some specific software that actually is targeted. Actually, there is a couple of different solutions for the molecular dynamics for chromatin. The question is very good because typically molecular dynamics is done for, like, say, for proteins. And in the case of proteins, actually, the complexity of the system and the molecular dynamics simulations are even though they are, computationally expensive, they are not impossible. But in the case of chromatin, if you try to do atomic resolution simulations, you will never get any result because of the number of basically atoms involved in forming double helix, which is 2 meters long. For a human genome, it's 2 meters long. So that's why you have to use coarse grain methods, represent fragments, segments of DNA, like 5,000 base pairs or 10,000 base pairs as a single molecules. The software that we are using is called OpenMM, Open Molecular Mechanics or Molecular Dynamics, and that is the basically very easy to use even in Python library to run molecular dynamics and molecular mechanics. So most of the so-called Spring model 3D models that I was showing was done in OpenMM, and we also have our own simulation code, which is Monte Carlo, which is written in pure C. So it's written from the scratch because that time we were not really finding a good solution for the to simulate our system.
Other questions from the online? Okay. So thank you, Derek, for this very interesting and enlightening lecture. And on behalf of ICDEC, I really thank you for being present in a virtual mode and gracing this occasion. Thank you very much.
Thanks a lot.
So this is the closer of today's session. On behalf of EC Department NEHU, I thank all the delegates, professors, speakers, faculty, students in successfully contributing to the second day of International Conference on Data, Electronics and Computing, ICDEC-2022. Thank you, sir.
There is a momentum for our session, sir.
Thank you.
And sorry, sir, I want to say something here about this.
Professor is that? Yes.
Yes.
Move to
Yes. Thank you, sir. I'm the organizers from this NEHU, and actually we were expecting you to be here with us in physical mode. Even from few days ago, someone from one official from Ministry of External Affairs called me that, is there any foreigner? I said that, yeah, one professor from Poland was supposed to come. We were happy, but somehow you didn't allow him to come. They said you didn't allow him to come. I said so that when I was driving, I received a call just recently, maybe on 6th or 5th, I've forgotten. Sorry, we could have felicitated you by at least 1 hour away of like Iburom. So we said, so, but because unfortunately could not do, and we are hoping you that in future we'll see you physically here in Nehu or somewhere nearby. Next year, at least, we are expecting that you'll be coming to Northeast and you'll have some more time to come over to Nehu and Shillong and Meghalaya. Thank you for delivering a nice talk. Thank you, sir. Good night. Okay, we close for the session today. Thank you, everyone.
Okay, thank you. So we close the session, Dariusz. So we'll be in touch. So thank you. Thank you very much.
Okay, thanks a lot. Thanks a lot. And a lot of greetings from Poland to all of you. Bye-bye.
Bye. Okay. Okay.